Hello, everyone. We are Group 1, and today we're going to talk about the history of smallpox. These are our group members. Next slide. Here we have the outline with the topics that we're going to discuss during today's presentation. We're going to start with introduction. We're going to talk about origin and spread, global impacts of the virus, scientific classification and structures, the genome, phylogeny, vaccine development, the virulation, historic biowarfare. We're also going to talk about the outbreaks and the clinical concepts and symptoms, the forms of transmission, vaccine evolution, eradication, the world after smallpox, post-smallpox drug developments, mechanism of action, and we're going to finalize with the conclusions. Next slide. So what's smallpox? Smallpox is an infectious disease caused by viral virus, VRV. As a member of the orthopoxvirus genus, the Varroa virus is related to other poxvirus, but distinct in its human specificity. It is estimated to have started 3,000, 4,000 years ago in densely populated areas like the Near East, Egypt, or the Indus Valley. Its catastrophic consequences on population caused it to spread around the world, affecting every continent and influencing the path of history. With a fatality rate of about 30%, smallpox is one of the deadliest illness in human history and has produced devastating epidemics all over the world. Next slide. So now we're going to talk a little bit more about the origins and spread. So China published the first account of smallpox in the 4th century. And then in the 7th century, India came next. In the 10th century, the Mediterranean and Southwest Asia were impacted as well. And during the 13th century, it was common in Southern Europe before beginning to spread over the continent and killing millions of people. Smallpox was prevalent in Europe during the 18th century, and there were notable spikes in the number of fatalities. Then, at the time, the causes and mechanisms of the disease were unknown. They didn't know how to treat it. They didn't know how it started. And the earliest methods were reported in the 10th century, and it was insufflation in China and cutaneous inoculation in India. And also to avoid the serious smallpox virulation was practiced, which they're going to talk about it in other slides. But it was basically consisted of inoculation with benign smallpox from the pustules of a patient. And then in Scotland, farmers, they started to attach wool soaked in smallpox pus to their wrist, and in some Italian mountain areas, the children were made to wear similarly soaked shirts. So they were just trying to find ways of preventing people to get infected because it was really like the fatalities were super high and they didn't know how to cure it. The results of all these methods were random and many people still died. Next slide. So for the global impact, before the eradication of the virus, in the course of several generations, for 1,000 births, 1/3 of subjects died between birth and the age of 1 year old, and 1/3 between 1 year old to 18 years of age. So it was responsible, the virus was responsible for 300 and 500 millions of deaths. And in the last millennium, it is estimated to have been responsible for 10% of deaths worldwide, which is crazy. After eradication was put in place, the world population increased to approximately 1 billion in the 1800s and more than 1.5 billion in the 1900s. Next slide.
So here we have a global distribution dated for how the virus spread out throughout the centuries. It is believed to have started either in Egypt, region of North Africa, or in Asia. It's not quite sure as to exactly where it started, but around that region. And from there, during the 4th century, it made its way into India, and like in around the 8th or 9th century, into parts of Europe as well. And it stayed there for a while. It took a while for it to reach North America through slave trade routes and trader routes for sugarcane, rums, other such things, and it made its way into the Caribbean, South America, Central America, and North America eventually. And there it stayed until its eradication. Next slide.
So for the scientific classification and structures, the variants of the poxvirus are the only virus particles that can be seen in a light microscope. The variants have a void or brick-like shape, like we can see here in the image, with a dimension of 400 by 200 nanometers and a linear genome of double-stranded DNA of sufficient length that encodes approximately 200 proteins. So here in the image, we can observe the structure of the virus. There are two subfamilies. There are the Cordopoxivarian, which infects vertebrates, and the Entomopoxvirus, which infects arthropods. Arthropods. Next slide.
Now, for the scientific classification, we are going to mention specifically what was the classification that affected humans specifically. As we mentioned earlier, there were types that affected vertebrates, but for this one, the one that affected humans was from the family Poxviridae of the genus Orthopoxvirus, specifically the variola virus. Now, apart from the major and the minor forms that are discussed later on the slides, on the research, they also discussed 5 type of forms, which are the main ones that have been discovered regarding the smallpox virus. So for the first one, it is the ordinary form, which is the most common one that affected around, well, that was found in 90% of the cases, and it is the one that has the fatality rate of 30%. Now, the flat form, it was present in 5% of the cases with a fatality rate of 97%. The hemorrhagic form was present in less than 3% of all the cases and had a fatality rate of 100%. The modified form was present in less than 2% of all the cases and had a fatality rate less than 3%. And the last one was the B sign eruption, which was present in less than 1% of all the cases. And so far, there has not been any known fatality rate for that form. Next slide.
So the variola virus genome consists of about 186 kilo base pairs. It is ended in covalent ends and which have inverted terminal repeats that enclose the coding region sequence. The coding region sequence is about 196 to 207 open reading frames. Specific to the variola virus, there are no open reading frames in the internal repeat regions, and 90% of the open reading frames match with other pox virus. The central coding region sequences contain conserved proteins, and the terminal CRS regions contain unique proteins that are specific to the host range and virulence of the different variola virus subtypes. Variola virus has about 3 clades if you try to put it into a phylogenetic tree. These are somewhat related to the 3 to 5 forms that people have researched and found major, minor, and intermediate versions. There is low sequence diversity identified among the different variola virus isolates, but the ones coming out of Asia in clade C are the most virulent, with clade A and B being a little less virulent. Diversity in clade C is caused by gene loss or intergenomic recombination. That was what they found trying to look for links in between A, B, and C to see whether or not they were completely related. And it looks like clade C is actually missing some of the end proteins that A and B still have conserved. Variola virus is closest to taterpox and camelpox. I believe the taterpox is from a gerbil that they found in Dahomey, Africa.
So in regards to humans, there are two main forms that we focus on when we talk about smallpox primarily, although there are more than two. But the two that I'm going to discuss are variola major and variola minor. Variola major is the more fatal of these two strains, which is also known as Asian smallpox, as it is believed to have originated in that region. Mortality rate is up to 40 to 50% on that particular variation. And last natural contraction was recorded in 1975. Variola minor is much less fatal generally in individuals that aren't immunocompromised. And it emerged in the 1800s in Europe, and the last natural contraction was in October of 77 in Somalia. So they're hard to distinguish between, this only not accounting for mortality rates. So in an event where there's only a few cases before an outbreak is stopped, it can be difficult to tell which particular strain was present. And a way that was developed to be able to do that was inoculating chicken embryos with whatever virus was present in the area previously. And based on how the virus behaved, they could determine which strain it was. This was before like the Human Genome Project and genetics was really a big thing in general. This was done like the 70s, so this used to be the best way to distinguish between them. And they found that the major strain was much more aggressive generally, and that the minor strain was much more susceptible to temperature change. Uh, it wasn't the best method I mean, at the time it was, but it still took 14 days to actually like get the results. So it really wasn't that great at the time. Next slide. So transmission is an interesting topic. Since the virus died out in the 70s, it is sort of not clear as to how it was transmitted, whether it be mucosal, uh, through skin to skin, or general like respiratory, like airborne pathogen. So they had to do a study based on like historic accounts in order to try and determine like how exactly most natural contractions happen. And based on the fact that variolation through either poking a hole in your skin with infected tissue, or the way that it was usually done was they would grind up scabs and blow it into somebody's no nasal cavity. Based on the fact that that would produce a modified, less severe case of the virus, they believe that it was not mucosal nor skin to skin generally. What they believe, based on the fact that most maidens at the time didn't contract the disease through coming into contact with like bed sheets and such. Based on that, they realized that the particles that were most contagious were probably so small that they couldn't be like lifted off the bed sheets after they had been thrown onto them, which would indicate that it was generally aerosol, so very small, fine particles that would get into your respiratory tract through there. And so that's how we believe that it was transferred from person to person, which would mean that airborne particles could stay in a room for up to 2 hours in the air. Next slide. And this is reflects what I mentioned earlier about the method of transmission potentially producing a modified version of the virus. So on the left, you see through dermal inoculation or nasal inhalation or variolation, in the case where they would blow it into your nose. The X on the individual indicates that mortality was much less common in those cases. Whereas if you contracted it through respiratory droplets, the result was much more severe. And generally what you would see in these like pandemic epidemic episodes in the history. Next slide. So the clinical course for the disease is quite long, lasts anywhere from 21 to 30 days, due to the fact that the incubation period varies from 7 to up to 17 days before the rash even, or before any symptoms even start to appear. But once you do become symptomatic, you typically have general feverish symptoms, aches, pains, and weakness for 2 to 3 days, after which the rash starts to appear. Once the rash first appears, typically the disease runs its course 14 days prior to that occurring. So within the first few days, you see your Manicupopular rash appear, and a few days later, vesicles appear after the rash starts. Pustulates then appear about 7 days after the rash emerges, and then if the individual survives to this point, uh, the scabs follow, and then scar tissue starts to develop, assuming the individual is still alive. The lesions reside deep within the skin, and they're pretty dense, have flattened surfaces on them, and generally most deaths occurred within a week too of contracting the virus. And scarring was very common among survivors. Next slide, please.
Now, there has been record of how this smallpox was spread around the world and its different outbreaks, although there's still discussion about whether it started in Asia or Egypt, since they have found evidence in both places. However, I think the earliest one was the one before Christ, 1157 year, which was recorded from the Pharaoh Ramses, where they found his mummy and he had traces of smallpox on top of his head. Now, from 165 to around 175, it was due to the Roman Empire, which was called the Antonine Plague, and this caused the pestilence to be propagated all the way to Egypt, Asia Minor, and Middle East through the Red Sea, and then to Syria, Chaldea, Mesopotamia, Lycia, and Sicilia because of the spread of the Roman Empire and the soldiers being um, sick with this virus. Now, the second wave that affected the Roman Empire happened in 191, and it caused around 7 to 8 million deaths. Now, from the 6th to 8th century, there was outbreaks in North Africa, Spain, and Europe. And in 1241, it happened in Iceland, where it decimated 1/3 of the whole population in the island. Now, due to the colonization and the spread of some of the major powers back then in the old world, the virus was able to spread to other places, such as the Puerto Rico, Cuba, and the Caribbean islands in 1518. And this caused the 50% of the indigenous population to die. Also, it was spread then in 1520 to Mexico. 5 years later, it was spread to Peru, which caused the death of 85% of the population. Then in 1560, it was spread to Rio de la Plata, and then in 1562 to Brazil. And then until the 1612, the settlers brought the virus to North America, which mainly affected Indians. And then from the all the way to the 18th century, this virus was still responsible for 10% of all the deaths in Europe. Then in the 20th century, it was still continuing to expand inside the United Kingdom, North America, and South America. Next slide, please. Now, there's potential for biological warfare um, regarding this virus, as there has been history that it was used before as a weapon, and this was used by the French and in the Indian Wars in the years 1754 and 1767, and it was used by the British in North America. Now, what also raised concern was the novel synthesis of horsepox virus, and it actually raised a concern because people started to think, well, scientists and all other people in power and responsible for this, that a terrorist could do the same with the variola virus. Now, why is this relevant? Well, this is relevant because the smallpox vaccination in the United States stopped in the year 1971, and 9 years later, in 1980, the vaccination stopped worldwide. So basically, we have no vaccines left. However, we have so basically we have two repositories that still have some remnants of the virus, which are in constant vigilance by the World Health Organization. It is also important to mention that these are highly secured. And basically we have one in the CDC in Atlanta, and the second one is in Vector Novosibirsk in Russia. Next slide, please.
So a very important part of smallpox is the concept of variolation. Variolation essentially is immunization or resistance building through the process of exposing oneself to either tissue or remnants of the disease. Not quite the same as the vaccination, but it's similar concept in concept. Variolation has been used a lot for smallpox as far back as 1000 BC, potentially. Um, it was used a lot starting out in Asia, where they would take ground up scabs from the virus and then blow it into the mucosal cavity of other individuals. And this would provide general resilience to the disease. It was actually pretty successful. And it did spread throughout the world, the practice, but it wasn't practiced as frequently as it could have been. And so it didn't really have as much of an effect on the fatalities as it could have. Next slide, please. Like I said, it was very effective when applied. And unfortunately, it wasn't used as frequently as it could have been. Uh, although somebody did take the concept of this and then eventually came to the concept of a vaccine, which I will talk about, I believe, on the next slide. Next slide, please. Yes, so this is Edward Jenner, who is essentially the father of the modern vaccine, many consider. Um, he himself was inoculated against smallpox and it was found to be pretty effective, even though it did for him have some pretty nasty side effects at first, which he didn't enjoy, but he still, despite that, practiced it himself when he became a physician and inoculated many people throughout, I believe, England and I think France as well. Uh, when he was on a farm later in his life, he noted that many of the milkmaids on the farm developed lesions on their hands, which were caused by a pox virus that was bovine in origin from the cows that they were milking. Uh, and something also noted was that these individuals with these lesions on their hands would never get sick when an outbreak occurred of smallpox. So, noting that, he decided to take a sample of one of these scabs from the milkmaiden's hands and inoculate a young boy named James Phipps with the pus from that lesion. And then he then exposed that boy to smallpox. And thankfully, it did work. If it didn't, that would be pretty tragic. And through that process, he eventually went on to publish a paper on it. It was not received very well at first, but later, upon him getting more data, it did sort of catch on in England. And that same strain of bovine pox virus was used up until like the 1900s as the base for the vaccine for smallpox. Next slide, please.
After Jenner's vaccine, vaccines have gone through a little of their own evolutionary history, starting with the first generation of vaccines. Dryvax is one. It was developed in the 1950s and grown on calfskin. And this was used in the smallpox eradication campaign. It was administered with bifurcated needles, initially where they would get like a small droplet of the vaccine and they would prick you on the arm with it. And some of the adverse effects included encephalitis, progressive vaccinia, and eczema vaccinatum. Progressive vaccinia is just a continuation of the disease outside of the pock area, where it would cause necrosis. And the eczema vaccinatum is similar, but it only affects the dermal area. ACAM 2000 and Aventis Pasteur are second generation vaccines that have been created to improve upon some of the adverse effects and also to improve like general manufacturing process of the vaccine. They consist of live replicating vaccinia viruses, and they are derived from Dryvax clones. They were licensed in the US, Australia, and Singapore and are currently stockpiled in the Strategic National Stockpile for the US and also by WHO. They are administered also using a bifurcated needle, but they do come with adverse side effects like myopericarditis and cardiomyopathy. Those are both heart conditions. Lister Elstree is also a second generation that was created around the 2000s, but instead of using Dryvax clones or calf skin, they were developed in rabbit kidney cells, and they were produced and stockpiled in Europe. These were also administered using bifurcated needles. Current third generation vaccines include Imvanex, Imvamune, which I believe is also called Genos. It is modified using Vaccinia Ankara, which is a less virulent variola virus version. Doesn't really have many adverse effects. It is a live attenuated non-replicating vaccine, and it is approved for use in Europe and Canada. That one is administered in 2 shots, which are done 4 weeks apart. And then we have the last third generation vaccine to be developed. Well, it was developed in 1957, but it was only licensed in Japan and is still only currently used there. It was derived from the Lister vaccine with the rabbit kidney cells, and it is a replicating attenuated strain. It is also administered using a bifurcated needle, but it doesn't have any adverse effects and it doesn't produce a POC region. And this one is currently being used for emergency listing for Mpox outbreak, according to the World Health Organization.
So after Jenner created his vaccine, his initial vaccine, and published his paper back in the early 1800s, he had the hope that he would one day see the world eradicated of smallpox. And while it didn't happen within its lifetime, it eventually did about 184 years later. Despite the pushback from skeptics and profiteers that would profit from the disease being around, there was an effort started by the World Health Organization to eradicate the world of smallpox. It was started around the 1950s, though sadly it took a backseat to attempting to eradicate malaria for a time. The stages of getting to this point, though, started in the 1850s in England, with awards being given out to healthcare providers that would provide the vaccine and also to people who received it in some cases, financial compensation. In 1902, France made vaccination within the first year of life mandatory. In 1966, the vaccination campaign from the World Health Organization began. Uh, and then in 27th of October 1977, the last natural contraction of smallpox was recorded in Somalia. In May of 1980, the World Health Organization finally declared that the world had been rid of smallpox officially. And it remains as the only human disease to ever be eradicated. Next slide, please. So now we live in a world post-smallpox. It currently only exists in 2 labs in the entire world. It used to exist in 4, but due to an unfortunate incident in a lab in Birmingham, a lab worker in an upper floor of a laboratory that had this virus unfortunately passed away due to it getting somehow into the air circulation system, and she passed away, unfortunately, after contracting it. She was the last fatality of smallpox. Now it's only located in 2 areas of the world, the CDC lab in Atlanta and Russian lab in Koskov region. Due to these being the only places where smallpox still exists, people are generally not vaccinated for it anymore. Old vaccines used to cause localized lesion, although some of the newer ones are better and don't, but it's still not practiced frequently in the newer generations. And so that's raised a concern as to if there exists a stash of smallpox virus that we don't know about, it could be potentially be used as a weapon of biological warfare, especially with how much terrorism that we see nowadays. It's not beyond the realm of possibility. And the U.S., especially and among other places, have sort of had outlines put in place to hopefully have like a rapid response in place where the healthcare workers involved in that would be vaccinated, and that would hopefully help facilitate a response that would be less severe if an attack did occur. Next slide, please.
So antiviral drugs have continued to be developed for smallpox in the case of an outbreak through bioterrorism. Some of the ones developed are tecovirimat or TPOXX. It is administered orally as a capsule and was approved for treatment in adults and pediatric patients in 2018. So it has been shown to be effective in variola, vaccinia, cowpox, monkeypox, ectromelia, camelpox. Um, so it's very effective, but it does come with minor adverse effects such as headache, nausea, abdominal pain, diarrhea, and things like that. So far, there have been 2 million stockpiled by the US in case of an outbreak, and it is currently undergoing trials for use in the monkeypox outbreak. Another antiviral that was approved is brincidofovir. Excuse me for the pronunciation. It's also called Tembexa. That one is administered orally and is either a tablet or an oral suspension based on weight. It was approved for medical use in 2021, and it does have a black box drug designation. Long-term use can lead to death, so it is not usually the first to be prescribed. It would be tecovirimat. It is potentially carcinogenic, and it has adverse effects such as gastrointestinal upset and hepatotoxicity. And it is currently being used in studies for cytomegalovirus. So the mechanisms of action for both tecovirimat and brincidofovir are in two different places of the viral replication. So tecovirimat is actually inhibiting the wrapping of the virion envelope before it leaves the infected cell. It interferes with the protein F13L and blocks viral VP37 protein, which is supposed to interact with Rab9 GTPase and a TIP47 protein, and reduce the wrapping activity. This prevents the cell from replicating as well. So brincidofovir is actually used to stop the DNA synthesis of the virus before it actually gets to the coding. The active antiviral is cidofovir, but it has a lipid tail attached, which is cleaved once it enters the infected cell. It is converted to the cidofovir diphosphate within the cell, and then this targets inhibition of viral DNA synthesis by inhibiting the DNA polymerase and acting as a nucleotide analog.
So, in conclusion, smallpox was a blight upon humanity for thousands and thousands of years, killing hundreds of millions of people.
Millions of people.
Oh, sorry. Did you want to say something, Melanie?
No, I was just saying millions of people. Yeah.
Oh, yeah. Thanks to a global effort headed by the World Health Organization, we now occupy a world that's free of that terrible disease. And the eradication of smallpox is honestly a testament to the human will, and hopefully proves that we can eradicate more, horrible diseases in the future. Next slide.
And also to add on that, for the conclusion, um, one of the main points that we took away from this research is that smallpox has different forms. Um, I think most of us only thought that a smallpox was only like one form and that's the only virus, and they all do the same thing, but no. So we have the two main ones, the major and the minor, but there are some extra ones too. So I think that's one key takeaway from this. And another key takeaway is that the lack of information that we have on smallpox is not due to not having the proper, the proper equipment. It's just that smallpox was around the times that it was really hard to study it. So, and basically, since now the smallpox is really limited, as we all saw that it's only available in like 2 research centers, that's why there has not been a lot of further discoveries regarding smallpox. And some of the research is also, really back in the day. So yeah.
Like, back then it was very outdated, and they didn't have the same resources as we have today. So it was really hard for them to understand the mechanisms and why it was spreading and how to stop it in the first place. They just didn't know, didn't have the means. Definitely. Yeah.
Yep.
So here we have the reference of everything we used, where we basically got our information from, and we're able to do a little research on the topic to understand more for the presentation about the smallpox and how it started and how it was eradicated and everything else. And then thank you so much for watching, if you are. I hope this was a very informative presentation with all the points that we brought out and everything we talked about. Hopefully now you have a better, whoever's watching, a better understanding of the smallpox. And yeah, I don't know if you guys want to add something else.
I think we're out of time. So
Yeah.
yeah. Thanks for watching.
Thank you.
You.